Today on Surfing the NASH Tsunami.
Probably the most long-lasting and impactful idea came from Vlad Bratsiu, who was talking about biomarkers and having faith in them. He somewhat tongue-in-cheek said you really almost have to form a cult to get there, in the sense that everyone just has to believe it, in the lack of the hardest of evidence. And I think what Vlad was really hinting at is the requirements for histology as the primary endpoint in NASH clinical trials is really rate limiting.
This weekend, over 200 NASH stakeholders have gathered in Deer Valley, Utah for the 5th NASH-TAG meeting. Join liver wellness advocate Louise Campbell, Global Liver Institute President and CEO Donna Kramer, and pricing and forecasting guru Roger Green, along with our guest surfers, NASH-TAG co-director Dr. Michael Charlton and Dr. Ian Rowe of the University of Leeds, UK, as they identify and debate major questions from NASH-TAG Session 3, all from their own unique perspectives, on the Surfing the NASH Tsunami podcast.
For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up. Our coverage of NASH-TAG Session 3, Season 2, Episode 13 of Surfing the NASH Tsunami starts now. As we speak, NASH-TAG is rounding the corner pole heading for home. If you heard my interview with Michael Charlton on our episode 10, he foretold that this morning's session would be special. Having listened, I can certainly second that sentiment. Speaking of which, and who's surfing with us this afternoon, we're delighted to have NASH-TAG co-director Mike Charlton with us for this episode. Mike, welcome to the real Surfing NASH, not just the interviews.
Thank you.
Joined by Ian Rowe, who is back from yesterday afternoon.
Hi, everybody.
And then Louise.
Good afternoon.
And Donna.
Hello.
Both back from this morning. Michael, that was a superb session you chaired this morning. We were chatting about it a little bit earlier and thought it was great. Since we have to be back in an hour and a half, we will work to limit this to an hour or less. So let's get started. If you've heard either of the previous episodes, audience, this is the same simple format. We'll start with Michael, who will give brief answers to the 3 questions we've been asking about the episodes about this morning. We'll talk about his answers, and then one by one, we'll work through with the rest of the group. At the end of that, we'll have a wrap-up, summarizing some key issues we've identified and discussed. And then everybody will rest up for Session 4 later today, although it won't be a long rest at this point. We will post this episode early tomorrow morning, Eastern U.S. time, and then we'll break, listen to the session, and then conduct a podcast tomorrow morning with Donna, Naeem Al-Khoury, and Jorn Schattenberg. One announcement, we're wrapped up with our Monday night lineup. In addition to having Vlad Bratsiu as a guest, Mazen Noureddin will be with us also, and of course, Stephen will be back. So with that, Michael, you're on the seat of honor, given that you are the co-director in the room today. So the 3 questions, and then I'll ask them all and then come on back through it. Question 1, tell us one point or issue you heard during this morning's session that you found novel or surprising, or that you believe will drive a different course of action. 2, what did the presentations in the session have to say about this issue that make you feel this way? And 3, what are you taking away about this issue that will lead you to do something differently or look at something differently? And with that, I turn to you and say, have at it.
Those are great questions. I agree with you that the sessions were excellent. I think probably the most long-lasting and impactful idea came from Vlad Bratsiu, who was talking about biomarkers and having faith in them. He somewhat tongue-in-cheek said you really almost have to form a cult to get there, in the sense that everyone just has to believe it, in the lack of the hardest of evidence. And I think what Vlad was really hinting at is the requirements for histology as the primary endpoint in NASH clinical trials is really rate limiting. And it's something which wasn't present for hepatitis C, for example. We didn't have histological endpoints. You treated the virus, and if you got rid of the virus, then it was presumed that histology and clinical endpoints would follow. We don't have anything as clear-cut as a hep C PCR, of course, but we do have other things. So we have indirect measures of hepatic fat with MRI-PDFF, which is shown to be a marker of changes in certainly hepatic fat content. Nobody would doubt that. And in some instances, inflammation and probably fibrosis. The predictivity for fibrosis is probably going to take a bit longer to emerge, because the drugs that have been studied to date were essentially natural history studies. So the drugs with selonsertib, simtuzumab, they ended up being very expensive natural history studies studies. And without much change in your endpoints, it was hard to test the delta in endpoints based on a biomarker. Now we have larger phase 3 studies that are with more effective agents like resmetirom coming through. We'll probably have more testable hypotheses for biomarkers in their performance of measuring histology. And more importantly, what Vlad was getting at was measuring or predicting clinical outcomes. We had David Pinata from Imperial College in London. They've already taken this leap of faith with oncology, where they have outcomes that occur more frequently and much quicker, with mortality, with dangerous solid tumors, hepatocellular carcinoma, cholangiocarcinoma, etc. So I think the idea that we would, as a field, really embrace biomarkers would have a profound effect on therapeutic agent development. Sorry for the long answer.
No, actually, that was crisper than some other answers that we get to questions like that. So you have nothing to apologize for. Vlad's been a frequent guest, and we were also impressed with Stephen made the comment this morning. He was now known as Vlad Verbose. So I was delighted that he actually hit his time points, as I'm sure you all were. Yeah, that was a pretty spectacular talk. But who would like to comment on what Michael just said?
I would. So I first of all, I want to compliment you, Dr. Charlton, for such a fantastic meeting. It's just really been edifying and rich, clearly very carefully curated and current too. Was very excited to see Vlad's full embrace of moving what we like to call is beyond the biopsy, and the campaign that the Global Liver Institute has had for well over a year now. So I would submit that Beyond the Biopsy is a better campaign name than the NASH-TAG cult. Just throwing it out there as a former PR exec. But welcome everybody to, and if you all want to walk at the front of the parade that I started, I'm happy to have you there. I think that it's important. I think that's what's been missing. And you're absolutely right in the language that this has been the rate limiting factor for the field. And that's why we had identified and started that campaign. And I remember being at New York investors conferences several years ago and being told, absolutely not, Donna, that's unrealistic. It'll be 10 years. You don't know what you're talking about. And now that, that horizon is shortening with fantastic, investment of time and intelligence and technology and resources. But I really think it is this realization that we cannot scale up diagnosis and treatment and management of NASH without getting beyond the biopsy. So I was very excited at the clarity of the points and the fervency in which he made them. The most startling thing for me, though, of the meeting was the NASH and HIV information. I honestly was not aware, and already have been with my staff to reach out to our HIV/AIDS colleagues and advocacy organizations to see how we can do the same type of outreach that we've done with our colleagues in diabetes. It was really striking to me, and I was really affected by that, and realized it's an additional way that we need to build out our NASH Council in the same way that we've integrated diabetes and cardiovascular organizations in making sure that NASH is seen as a very big tent, is indeed a public health issue with intersectionality amongst all these conditions.
I was listening to HIV and I had kind of a similar reaction, but the real reaction I had was, boy, I'd love to hear what Donna has to say about this. So I feel like we've achieved something already.
I was busy. I was slacking my staff saying, I need to reach out to these 3 organizations right now.
But we've all been hanging out entirely too much, I think, but it's mostly a good thing. Louise, why don't you go next? What did you take away?
I think Donna's sort of said most of the points that I was going to make in that context. And I think the actual presentation that Vlad did was amazing in the context that we just have to prove realistically that everything we're doing proves that trend down. And I thought he had a very good argument for that, where we see the improvement. Do we have to be absolutely precise? Well, we're up against biopsy, which I think has been shown again not to be the most precise mechanism. So I think by going beyond the biopsy, by going non-invasive in lots of ways, the ability to recruit more and more people in a quicker timeline is now a reality to be able to assist the development of most of these medications. But I think the one by David Pinto, he showed some really, really good examples of the platform trials in a real way of being able to show how they do work within certain areas. And I think I commented this morning on, can we now look to split steatohepatitis cirrhosis improvement from NASH, fibrosis improvement. And basically what he did was show those 3 separate markers as individuals rather than the combined that we're using at the moment. And I think there was also another section, I think it was Vlad or someone else, that basically also supported Manal's argument. It was the diabetes section and the medication, that non-progression is an outcome of that medication and it's a good outcome. So I think a lot of what's happened today, that's down to the great way that this has been choreographed, is we're tying all of the loose ends together in a holistic way. And I think that's really, really nice as we move towards the closure of the conference.
It's funny when you think of what the FDA's purpose is, which is for any disease to identify drugs that can improve how a patient feels, functions, or survives, then they should really have that at the front of what they're doing. And they've already taken the leap of faith that they'll accept a surrogate biomarker for the clinical endpoints, which are worthy goals to improve those hard clinical endpoints. So if you're already going to accept a biomarker, which is imperfect in ways that everyone recognizes, which is liver biopsy, why not go a step further and take something that will make it much quicker and probably superior in terms of efficacy when you can evaluate multiple agents in series and in parallel by taking non-biopsy-based biomarkers instead of the biomarker which biopsy is. So I think it doesn't take much, or wouldn't take much, for the FDA to get its arms around non-biopsy-based biomarkers, particularly if they really consider what their mission is.
Michael, I have a question before I turn to Ian, which is, I'd never seen discussed in public before the idea that you could do away with 2Bs entirely.
It was great. I loved it. That's actually my favorite single part.
And that was, to me, that was the moment that summed up the morning, because I'm a fan of saying dare to dream. But that was the slide that hit me with, if you dare to dream, this is what you can achieve.
The whole talk should have been titled Not to Be.
Yeah, fantastic. Not to be. There you go. I like it. Ian, what do you think?
Well, so I thought Vlad's talk was excellent. And we talked a bit with Manal last night about a biomarker-driven trial design. And I think it made me think of a couple of things. One was the issue that first brought me to the podcast, and that is what the market for drugs in NASH actually is, and what do we want a therapeutic to do? Do we want a bespoke boutique drug that's going to be used in small pockets of the population in a highly personalized way, or do we want something that is widely applicable, easy to use, and reduces mortality across the population? And my argument's always been that we would be after the latter, and a biomarker-driven strategy definitely fits with that much more easily than a biopsy-driven strategy. It would be a real step forward to get to that. There are a couple of issues, though, that didn't come out in the discussion, and those are around the population that you choose and what the endpoints actually are to, from a sort of clinical perspective, to get the proof that you would need that the biomarkers are really measuring what you want. And some of that comes down to what Vlad was saying about, requiring a biopsy to prove progression to cirrhosis, which I think that's still in the mindset that you'd be recruiting patients who were pre-cirrhotic. But actually, if we're going to throw design up altogether, then you'd be as well to say, well, what we should really be doing is to be picking those patients who are at risk of liver-related mortality with compensated cirrhosis and F3, putting them all in a big pool and treating them. That will make the trial structure, the trial design and implementation much more practical and applicable to real-world practice at the end. And I'd be interested to hear what Michael thinks of that and some other thoughts too.
Yeah, so I think the important point of Vlad's idea of doing away with the 2Bs is that you still get your hard clinical endpoints that FDA is rightly concerned about, but with an accelerated development program up to phase 3. So I don't think anything is really lost. I mean, I think worst case, you may have agents with marginal benefit that are discarded pretty quickly because they didn't meet biomarker standards, but that's okay. The companies would be free to go and pursue those through more biopsy-based studies if they wished it. So I really don't see an important downside to it. I think this is an idea that is worthy of further consideration, maybe as a single topic symposium.
I heard that in your question, but I also thought in your question, Ian, I heard about who the patient target should be and how broadly the net should be cast on that issue also.
That's right. And I'm a hepatologist. I'm in the business of preventing deaths from liver disease. Well, I guess we can come back to what the overall possible benefits of treatments might be in the future. But if we're going to do that, then we've got to be picking the population that's at greatest risk. And at the moment, we're a little bit away from that, I think, in the vast majority of cases.
Take it back to the FDA for a second. Michael, we noted on last night's podcast that you would put up Larry Kramer as your last slide of the morning speech. Part of the question being who will be the Larry Kramer for fatty liver. But I want to talk about Kramer in a different context, which is that the FDA is a public agency and it tends to be guided by the last set of politics that really got in its way. So when I came up in this industry, people were living in the aftermath of the thalidomide horror of the early 60s, and the agency would prove nothing. And then the big thing that Kramer did was change the entire thought process. That, it was Louise, Kramer's position was Donna's comment that you love to quote, which is, if I'm going to be dead anyway, why do I care if the drug is toxic? And to do that, as I said yesterday, he had to destigmatize homosexuality so that we could understand that we were treating HIV patients like any other patient. And then after that, he had to go on and make the argument for we have to do things in different ways. Now, since then, the yaws have been somewhat less dramatic, but I think the agency's general position, and as I was thinking about all this today, I was thinking back to the FDA webcast, is they will take great risks for fringe populations and populations at immediate peril. And they are unwilling to take those risks for larger populations that they see as being at less peril. I'm not saying that I see the fatty liver community as being at less peril, but they are larger, and except it's a less immediate kind of a thing than HIV was or some of the rare diseases they're aggressive with right now. Go ahead, Donna. I didn't mean to cut you off.
Sure. No. I was so eager to jump in. One thing is that, as I've been a student of social movements and activism, is that I really admire the HIV/AIDS community because they got very smart and they're expert. Created expertise within themselves on a variety of different subjects, regulatory being one of them. And so I think it's important that we, see liver health advocates across the ecosystem. So in as much as we're involved in legislative activity, and we're all over the hill in the past few weeks and continue on throughout the year, and we've been talking with CDC and made some very simple asks that they granted, and it was as if no one had ever asked them before. And with FDA, we have been very persistent in communication that they had not been treating NASH and liver diseases as a whole with the urgency with which either clinically or morally we felt that they should. And I think that we certainly feel, now that we certainly don't want to be confounded in any attack on the agency. I've been an FDA patient representative for over a decade. I've worked and continue to work very productively with probably about 5 past FDA commissioners. So I think in this new year, we've been able to draw together the hepatology, nutrition, and the cardiovascular renal division staff together in one meeting, because our request had always been to view NASH in particular and liver health overall from a interdisciplinary or cross-disciplinary, cross-division point of view, and to make sure that they were utilizing all of the tools and all of the resources within the agency, and were also requesting outside expertise appropriately. And so I do feel that we have a certain amount of momentum within the agency. I think particularly when we showed all of their recent actions across liver diseases, and when you paint that picture together, it's quite striking in how they had not been treating liver diseases, whether in cancer or NASH on the drug or the device side, or hepatorenal syndrome, or any of these things, with the regard that they should and that the data supports. And so I do feel that we will continue to be vocal and persistent and clear with them. And I invite everyone to join us. So yes, I am less of a chain myself to the gates type of advocate than you will see me again and again in the past at White Oak or 3 times a week at whatever event you go to in Washington. And so you will just have to do it, because I will always be there, and I will always be asking for the same thing until you grant it. And so it is more of a Washington way, but I think it can be effective and perhaps more suited to what we need to do to bring liver patients across in terms of the recognition and the reduction of stigma as we're building this larger community for them.
I think Larry Kramer, was the perfect advocate. So he was highly articulate. He was a playwright. He was fearless, and he had it was his job to be theatrical. He knew how to get people's attention, and it wasn't by just being persistent. It was also by being dramatic as well. So I think needing someone like Larry Kramer is an important unmet need at this point. The conveyor belt of illness for HIV and AIDS, it was not a huge conveyor belt, but it was short and it got people to death quickly. Now, NASH, the conveyor belt is much longer and slower, but there are far more people on that conveyor belt, and just as many, probably more, are ending up dying at the end of the conveyor belt. So the FDA has to be shaken out of its conservative natural state or instincts, which is where they have retreated to. Ironically, the Liver Forum, which has been a major source of engagement between the liver community and the FDA, was started out of HIV and Hep C interaction as well, and fatty liver as well. So it can be an effective way of interacting with FDA. I think it has really failed in the context of fatty liver disease or NASH. There's no reason why it has to continue to fail. I think they have the ear of FDA, and they just have to tell a different story and make them think differently.
I think my point is that you need the multiple types of pressure. So you need the policy wonk and the data and the drama. And so, GLI is putting together our honorary advisory board and talking increasingly to find, as I referred to on another one of these, our Liz Taylor moment in HIV, when Liz Taylor embraced Rock Hudson and there was sort of a turning point in the thinking, in Hollywood. There are many other moments, of course, but that was always one that was a striking point for me. And so, as we have tried to bring the liver community together and focus and build a larger tent and just, pick a color, burgundy, pick a month, pick a day, come together and use one voice and one momentum and overcome some of the fragmentation that, we've seen in the past. I think we can be stronger together, in sort of a full court press and surround sound and bringing the multiple aspects of what have been contributing to successful awareness campaigns in other therapeutic areas.
So a couple of thoughts about Larry Kramer. And although I said I wasn't going to do that again, sorry. And also about the nature of the population that we know of, but maybe aren't always talking about. The two great books about the early post-Stonewall AIDS community were Randy Schultz's book from San Francisco and the band played on, and Charlie Kaiser's book from New York, Metropolis. And both of those books, particularly Metropolis, made a point that Larry Kramer was really hated by the entire gay establishment. They wanted nothing to do with him. They were throwing him out of rooms. They were throwing him out of meetings. They threw him off the boards of organizations until the head of the gay men's health crisis, as he was dying of AIDS, looked at Kramer and said, "Keep going. You've got it right. We had it wrong." So if he hadn't been willing to break so many glasses and break so many eggs, I don't know that he would have had the effect that he had, which, Donna, isn't to belie your point. It's one of those that takes all sorts to make a world kind of comments.
I think that's why, Wayne Eskridge is such a wonderful voice. Because unlike me, he's not a creature of Washington. He's not a lawyer. He just, says it from his experience as a patient. And sometimes it can be like I think about it, so the Goldilocks theory of advocacy. Some is too hard, some's too soft, and so you end up just right. And so, as we saw in the, hepatitis C access advocacy, there were some organizations that literally had websites with things burning on fire. It was too hot. There were some that were perhaps, too cool. And so it ended up, I think, with some folks in the middle who were able to work and keep pushing and pushing until we got access for the broadest class of people with hepatitis C. So I do think it takes different types of expertise, different types of temperaments and voices and skill sets to bring this across the line. I don't pretend to have all of them.
Ian walked down a path a moment ago that I want to walk back to medically and in this way, which is, so what do we do for cirrhotics, right? It's even harder to get drugs approved for them. There are no surrogate endpoints. There isn't as much money in it, so the advocacy from the drug companies isn't going to be there the same way. Ian, why don't you flesh out the point a little further in terms of all the different dimensions, medical, social, et cetera, and then let's spend a little bit of time with it, because I think it's a great question.
So, it comes back to this sort of perspective and comparison that I made last night about development of drugs for cardiovascular disease starting in secondary prevention, where event rates were going to be high, so populations could be relatively small. And I say relatively, the first statin trials had 4,000 patients in. And understanding that if you had that sort of scale of trial with event rates that were significant, then you could, if you had a drug that did even relatively marginal benefit, then you would be able to observe that in a relatively short time frame. And it's always been a bit of a puzzle to me as to why we haven't started there. I know why we didn't start there in hepatitis C all those years ago is because the drugs were completely ineffective and, poorly tolerated and not that safe. But once the DAAs came, we could have headed off all of the concerns that Cochrane subsequently came out with a very short placebo-controlled trial over the course of probably 6 months that would have shown clinical benefit. And doing that now, learning those lessons and doing those studies now without liver biopsy with clinical endpoints, to me makes complete sense. And I think we're beginning to see drugs now that have substantial efficacy that would probably be worth testing early in that way. And some of those studies are going to start, so semaglutide and cirrhosis, but other drugs are coming too. And if we could do that, we can show benefit, validate surrogates in terms of non-invasive tests in cirrhosis, then the path will be a lot shorter that way. Maybe it carries a bit more risk for a company going into that arena, but from a patient benefit perspective, I still think that's the quickest path.
I was struck by Michael's point about the FDA changing their mindset. I just wonder today if TB medication and injection was coming through the FDA, whether or not they would approve any drug with 100% scar ratio, because obviously that is one of the major side effects that we live with once we've been vaccinated for TB is a lifelong scar. And the risk-benefit ratio, the FDA were very keen on when they did their discussion for NASH as to exactly what do they want to define as risk-benefit ratio. Patients are far better at defining what risk and benefits they want, and that they need to be part of that discussion. But could we have moved too far that we prevent excellent medications because we're too tentative or too stuck in our ways and our own definitions of what's good and what's not so good? So I think the other thing is that it's now hepatologists in the nutrition side, and I think there is a difficulty sometimes to move hepatologists to different points of view or away from particular values that they hold, and biopsy is obviously one of those, and they were still very keen to hold to biopsy recently. So it'll be interesting to see if blood can move to beyond the biopsy. I think the FDA certainly can.
I think that's an excellent point. We wouldn't have aspirin. There's an almost countless number of medications we would not have if we went through the same approval cycle today. We almost have to have, I think for the national debt, there's this clock that spins with the wheels of the amount of the national debt. We need to have patients dying from fatty liver disease. I had a young mother, she's a Hispanic woman, who we saw progress from compensated to decompensated cirrhosis. She had teenage children. She developed a portal vein clot, died following a TIPS PVR attempt. There are patients like this all over the country that we're not able to get therapy to when they're compensated and amenable to therapy because of what's thought to be a safe and conservative approach, in reality is a cavalier and dangerous approach because of its conservatism. So I think we have to really have the FDA think about this in ways that it does not come naturally to them.
I certainly agree. I think Louise and Michael, you articulated it very well. I'm happy to share the communications we have made to them, and you clearly need more support and heft from the community in that. I remember preparing for a meeting with FDA, and one of the patients, one of the advocates that we wanted to bring with us could not attend because she had developed liver cancer. And we did let them know that. And so I think that's an important part. And I think as we plan for International NASH Day on June 10th, having that, just like, the president took out of his, pocket the number of people who have died from COVID, the number of people who are dying every day from NASH, learning about it just when they're diagnosed with cancer or transplant when it's too late to do all the things that we could do. We are not treating it with the urgency that people are depending on us, all of us, to have. And so any ways that we can make that point with greater clarity, we welcome.
I think that's right. And I think if we think back to the Arnold conference, we covered the James Pake issues when he was talking about metabolic disease, and the mortality was something like, if I remember correctly, 360 per 100,000 if there was a metabolic associated cause, because of course he went on to prove that high sugar was the biggest cause. And I think the figures were astronomical if we look at the per 100,000 death rate of COVID, and you then superimpose that with the stuff that James Pake presented at Arnold. It's quite startling how many people are dying of NAFLD and NASH.
This is really difficult for hepatologists because, I work in a transplant centre. I do a clinic for patients who've got HCC, and all I see is complications of liver disease, and it's there in front of me all day, every day, and it's horrifying. And the light that we have in viral hepatitis has just not been It's not been there for really any of the other diseases, and it is really hard. But I do have some sympathy with the agency because we've selected a population for trials where the risk of liver-related mortality is actually quite low. If you take patients with F2, F3 at 10 years after diagnosis, maybe 5 will be dead from liver disease, but 15 or 20 will be dead from other causes. And it's unfortunate that the first drug that we took them had a liability with, the potential for an increase in cardiovascular disease, which is the number one cause of death in that population. So it's almost a perfect storm of a drug with little efficacy and a very concerning liability. I think LDL is causal in coronary heart disease, and to see that signal that's not controlled with anti-lipidemic medicines, I can understand their perspective, particularly with surrogates that are unvalidated. And I come back to the point about selecting people with more advanced disease to enter trials, show the agency the benefits, validate the surrogates, and move on. Develop better drugs and improve the outcomes for our patients.
I agree. I think that sometimes we conflate meeting the needs of patients across the spectrum of NAFLD and NASH with approving a drug for everyone across the spectrum. And so I think that you're right, that we need to find a population, a patient profile for which there is clear benefit, and the benefit risk trade-off makes sense to regulators, clinicians, and patients. And get a drug across the line. And as our knowledge increases, we can then build out to appropriate populations. We've talked about it as having, a solution for every stage. And so sometimes the solution for those earlier stages is not medication. And I think we've preserved our credibility as an advocacy organization because we've never maintained that the solution for earlier stages is a medication. It's going to be connecting to lifestyle intervention, dietitian. It could be bariatric surgery. It could be, for the vast majority, it's not going to be a drug. And so for those who will need, a medication in addition to lifestyle, then let's try to, get those subpopulations and subgroup analyses a little better so that we can, enrich and power trials so that we can show appropriate effect.
The sweet spot is probably stage 3 patients because that's where the clinical events and the mortality and morbidity will come from. And the impact of fatty liver disease, I think, is more substantial than it may seem. So nobody would probably argue that 50% of HCC is not related to fatty liver disease, and people rapidly die from HCC. You've got about a 20% 5-year survival rate from HCC. So Ian and I in our clinics, these are the patients that we're seeing, but we're not seeing the other side of the iceberg so much because they're still in primary care or not in any kind of care. So identifying a subpopulation, I think a stage 3 or likely stage 3 or a higher ELF population, which is where the clinical events happen. I think you could design a study that would accelerate development, give patients, like the lady that I described, something to offer when they're still compensated.
First of all, as I've been listening to this conversation, I keep thinking compensated cirrhotics over and over again. I take your point about phase 3, but compensated cirrhotics are the point where everybody feels the people in that place are on the knife's edge. I remember 10 years ago, I was doing some work, talking about doing some work with Vertex for their Hep C agent, and their general counsel looked at me and I said, so tell me the story. He said, the bravest people in the world are reformed addicts. And he started in this very emotional place where he said, number one, they live with every day with their past. Number two, because they self-medicated, it probably means that what's an easy emotional life for us to live, they can't. They've got the pressure of this fear and we're not helping them. I thought that was amazingly gripping. And part of what I'm hearing in this conversation is you could say the same thing about reformed alcoholics who are compensated cirrhotic. And I'm sure there are bunches of them in Hollywood who are in dramatic places where you could make good out of that. I'm looking for where the emotional edge is, and that's the place I intuitively go to. Michael, I think you're right about F3 is where the patients are, but a lot of those patients will drift over into compensated cirrhosis, alcohol or no alcohol. So that's not so far from the knife's edge. I think earlier than that's a problem. I do want to throw in one more in some ways, completely unrelated point about things the agency isn't thinking enough about. And this was the last point I made after I excused all you guys from the phone call this morning. But, we're worrying right now about having histology around for drug approvals for 5 years, at least. Well, that's nice. But in probably 2 to 3 years, the agency will approve a drug that can be widely used for patients with NASH. Those patients are not going to get biopsies because doctors aren't going to give them to them, even if, in fact, that's what the labeling says. And if at that point we've not made yeoman progress in figuring out who needs this drug and who doesn't in the absence of biopsy, we're going to be creating all kinds of problems for ourselves. And because we're so caught up in this issue of getting the first drug to the finish line, I don't think I've heard a lot of conversation about the day after. Are those just conversations I don't hear? Or Do you think it's easier than I say, or is there a point in what I'm saying?
No, those are conversations that, particularly before last July, we were very far along on with ICER on value, structuring a payer summit and having conversations with many about, would biopsy be required, and in-depth conversations with them and presenting them with data and getting from them what data they would need. So I think that, at least from the Global Liver Institute, point of view, and our NASH Council is now pretty broad and does have participation from the major patient voices in NASH very actively. I think that we've sort of reset. So there were things that we thought were moving so quickly that perhaps we didn't need to do. And so we're putting those building blocks in place this year, such as a patient-focused drug development meeting to get at what do patients really think are the benefit risk, trade-offs, and how do we think about it, and how should the agency, view those from our informed perspective. I think it is putting back into place, these conversations with payers now, with new cost effectiveness data, more data now that we've also changed ICD-10 codes, and can get greater granularity into, the stages of NASH and in the, of patients in the EHR, and really understand what is going on with different stages of NAFLD and NASH populations within different health centers. And so I think that going back and making sure that all of those boxes are checked, all of that groundwork is done, all of those stakeholders who will be pulled through for adoption of a drug are as prepared as the regulators are.
That all makes sense. I get that. I'm wondering if there's any way that can be leveraged into the other discussion about histology. If we're going to need a standard that lay hepatologists and associated specialists are going to need to figure out when to prescribe a drug for a patient, is there a impetus there to move the entire beyond the biopsy conversation further, faster? And can you then drag the FDA along with you? Just a thought. Could be completely off base.
Once the drug is approved, it'll come down to the payers, Roger. And we've had an increasing number of advisory boards with payers representing some of the larger payers in the country. And they're aware of data from Kaiser, for example, with 2 million patient study, that 1% of people with an ICD-9 code of fatty liver disease of any kind have ever had a biopsy. And only 5% of people with NASH cirrhosis have had a biopsy. And those numbers aren't getting larger, they're getting smaller. If guidelines from ASD, for example, don't indicate that a biopsy is necessary to start treatment, then I don't think payers will require it. They didn't require it, perhaps some of them did initially. They limited it to biopsy-proven F3, F4. That rapidly dissipated. And I think you'll see the same thing with fatty liver disease once we have a therapeutic agent in hand.
Can I ask, because I don't know the answer to this, and I keep asking, and I'm yet to receive an answer that I understand. In that context, so let's say I've got a patient who's got diabetes, got BMI of 32, and he's got a FibroScan that measures his liver stiffness at 12 kilopascals. And I think, in that post licensing and approval world, that I want to offer him treatment. And the treatment's main benefit is on liver injury, so on NASH. How do I gauge what the efficacy of that treatment is going to be for that patient without a biopsy?
Do you mean on treatment, Ian? How do you gauge efficacy once you've started therapy?
Yeah, or up front, how do I know that drug's going to work in the way that it worked in the population and the trial? When I know that if I put that patient forward for a study following a biopsy, that there would be a greater than 50% chance that they would be a screen failure.
Well, I think the patient pathway is that there are only three possibilities. That one, you repeat your FibroScan, let's say a year, could be 6 months, it's just for the sake of argument, a year, and it's either going to be the same, lower, or higher. If it's the same, you could say, well, it would have been worse. It could have been worse had I not given drug X. Okay, and if it's lower, you'll say, great, it's working. And if it's higher, you say, boy, I maybe need to double the dose or change to something else or add a second agent. But these are anticipated to be multi-year metabolic style therapies, I think.
Yeah, so that bit I understand, how I gauge what happens when I put them on treatment. What I don't understand is now I've got 10,000 patients or 100,000 patients. If treatment response isn't the same as it is in the trial, that has a big impact on cost effectiveness. That's obviously a lesser concern, I guess, in the US system, but it'll be a big concern in the UK. And I don't see NICE approving us to use a drug that was based on a biopsy-based strategy without biopsy up front. And for European countries, I think that will definitely be an issue.
Right. That's yes, I agree. That's what I've been pointing that out for. I guess I'll speak up in the future. That's what this past few days has told me, that I'm perhaps too shy, that I have not been as visible or as vocal as I need to be, or that the Global Liver Institute's, work and initiatives have been perhaps shining our light under a bushel. And I pledge to do more to share it with all of you so that you can give us feedback on how to amplify it and participate in it.
We have about 10 more minutes left in an hour, and we spent most of it on this set of issues. What else in the session should we be spending time talking about that we haven't gotten to yet? We talked a little bit about the HIV. We didn't talk much about Quentin's paper about the alcohol mix patients. Maybe spend a little time on that, or is there anything else that was in the hour that you think we should cover?
Quentin's paper was great. One of the shocking numbers he put out there was that 60% of people consumed alcohol in the last year. I'm hanging out with the wrong people because I'm going to say it's higher than 60%. I was surprised by how low that is. So this is going to be an issue. And they're often excluded if you state that you're drinking above a certain amount. You're often excluded from drug trials. But Quentin, I think, raised the question, maybe we should be including moderate alcohol, regular alcohol consumption in clinical trials, because that's what's going to be out there in the real world. 60% of people are going to be regularly consuming alcohol. So I think maybe that's something we should consider going forward, is bringing them into the trial fold and stratifying, but it adds expense as well. And it may affect efficacy in a way that negatively impacts outcomes. So it's a thorny issue, but I think it's important.
Do you think it will negatively impact outcomes, or do you think it will skew by positively impacting outcomes? When we've added FibroScan post anybody admitted with alcohol as a cause, a considerable number of people have changed their behaviour because they didn't appreciate that they had any level of fat or any level of scarring. And we use it, certainly at Imperial, we were using it nearly in every patient that had been admitted to the St. Mary's site was getting a FibroScan. And it was amazing how much behaviour we changed. Some of these patients were attending the emergency department 10 times a year, and we reduced that to no more than once. So people really took back. And I think the opportunity of adding different levels of alcohol to a NASH trial is actually you treat the underlying behaviour because you're engaging with people. And I think that's what we see in the placebo, is this engagement. It might not be the outcome of the study, but you're going to really positively skew the data because the fat will drop. We use it in detoxification by watching liver fat drop. So it does add a new dynamic, but it's real life. People drink.
That's an excellent point. And if you think of separate alcohol, there's a nutritional side. People who come into clinical trials may be getting good health care, certainly in the United States, sometimes for the first time in a very long time or ever. When we had a clinical trial that was multi-center and multinational, including this duodenal mucosal resurfacing, the Brazilian arm of the study, the placebo response rate was 3 times what it was in North America. And we think the reason for that was they were getting access to medical advice and care for the first time in a while. So the placebo response rate may reflect drinking less in a way that you wouldn't do if you weren't engaged in a clinical trial. So I think that may be one of the downsides to alcohol, is that people will take advice, and for those first 12 weeks at least, they'll drink a bit less.
I was going to say that would the data be cleaner if there was a certain run-in period where you then, people who do, sort of respond to just the, lifestyle intervention, attention, care, then are kept in care. Now we have them, linked to something, but they don't proceed into the trial and sort of confound the medication effect.
It's exactly what I would propose is a lead-in phase for the study, for clinical trials in general for NASH. I completely agree with that.
Yeah, and I think in general is probably right? Given what you're talking about with nutrition, diet, and relationship between diet and placebo rates at the beginning, lead-in sounds like the right idea, period.
Meeting Nagashalasani's criteria for things that predicted F2-3-4, it was methionine, choline, and PUFAs, and you only need a 4-ounce steak of fish, which is not that huge, 5 days a week to meet those criteria. So if you modified your diet just enough to reduce your carbs to less than 48%, and a fish steak 5 times a week, you're there. And that's not hard advice to follow.
Assuming you can afford to do that. I think you get to certain socioeconomic levels where that might be harder than it sounds. One
thing I did want to make sure that we did note before we wrap this up was I appreciate the clarification that the term baffled isn't necessarily helpful to the current discussion and design of trials, because it does sort of conflate these issues of, alcohol use and others. So I really look forward to to us. I'm not wedded to the name of NASH. I didn't come up with it. It's helpful to some patients. It puts some off, but it's what we have now. And I do look forward to conversations about, how we evolve the terminology as our understanding of the different types of NASH there are. But I think it was really helpful for where we are now for Quentin to sort of weigh in that the reasons why he felt that Baffled is not useful to our work right now.
I'm trying to remember the acronym that Naeem Al-Khoury gave us for why he uses Baffled. I think it was bilateral or bi something.
Both alcohol and fatty liver disease.
Both alcohol and fatty liver disease. Yeah, both would be simple, but I thought Baffled was exactly the right place to start.
But you might not have alcoholic. Some people don't drink.
Right. And the only question I had, Mike, about the 60% that don't drink is I know I don't hang out with the wrong people to that degree. So I was wondering how many folks were lying to the survey takers.
That's a fair point. It's a fair question.
If anything, that makes taking alcoholics out of the studies even more damaging.
Roger Williams had a fellow from Australia come over, and he sent this fellow around to meet with everyone who had had a liver transplant for alcoholic liver disease, and then asked them if they'd had any alcohol since their liver transplant. And when they came to the clinics, about 1 in 5 people were saying yes. When this fellow went to their households and sat down with them, 80% said they were drinking. So I think your point is well taken, Roger, that those numbers on the basis of a survey or somebody face-to-face asking you whether you did or you didn't, I think a lot of factors will come into your response.
Probably right. Anything else we want to touch on before we close?
I just wanted to say one thing, that I quite enjoyed Marianella's presentation about the phenotyping and becoming more and more predictive of who's going to be the rapid responders. I thought that was actually a really interesting set of topic that she presented. I
completely agree. It was a very clean, thoughtful presentation.
Yeah, I agree. When we talk about the economics of disease over time, right, the rapid progressors are going to be the people where the most money is spent. So anything we can do to figure out who's likely to cost the most in the short term is a step in the right direction in itself. And then if we can start to figure out how that might connect, how the pieces might fit together, I think you're right. The economic model becomes a lot more compelling. Thanks, Louise. I meant to mention that talk, and it slipped my mind.
Each of them were fabulous today. That's why it's hard to mention them. The mini debates in diabetes were, fantastic and very helpful. And clearly, the NASH and diabetes population is such an important one to talk about. And even the questions that help delve in deeper to getting away from from BMI and looking more at visceral adiposity, lean mass versus fat, and then where the fat is. We had sort of a funny moment in our GLI Live last week with Dr. Carr from the University of Pennsylvania, and we were talking about, what could be some of the sources of protective effects in African Americans, and part of it was where the fat, becomes deposited. And so since the show is for more laypersons, I sort of summed it up, at least for African Americans, that perhaps it's thick thighs don't kill you, but stress does. Where the fat goes matters, and what the body composition is seems to matter. And I look forward to more conversations.
That's great, Donna. As a guy who's lived with fairly broad thighs his entire life, I feel immediately edified. So why don't we do this? The question we've used so far is, what are you most looking forward to the rest of the meeting? But there isn't that much meeting left. So what I'd like to ask each of you is one way that you think what you've learned and are learning at this meeting is going to affect how you do what you do when you leave, when this meeting ends. Ray, you want to go first?
I have a list of takeaways. Certainly, several of you will be or have already received invitations to participate in International NASH Day. Clearly, I need to send some folks are beyond the biopsy campaign materials. We're reaching out to the HIV groups as we speak. So those are my 3 to-dos so far.
I think I'll jump in next. I think the evidence is mounting for non-invasive techniques and the potential of interventions, brief intervention, anything with FibroScan. It's not the best, but it's actually a very good marker, and it's a very good motivator. And I think we've seen more evidence today with the HIV population, and just that short study, you were able to assess steatosis. And obviously the technology within FibroScan has improved in the last 12 months quite significantly. So what I would certainly like to see is a lot more of these studies now say whether they were using continuous cap or whether or not they were using smart depth, because we know that those 2 parts of FibroScan make it even more of a stronger marker now for the trials going forward. I don't think we can judge the current FibroScan technology on a lot of the evidence that we've had in the past. So I think as we move forward, FibroScan will become stronger in those areas, and I think that's what we want to see.
This has been very thought-provoking. meeting and the discussions. And the thing that I think is greatest for me is trying to get a way of identifying those patients that we've got who are at greatest risk in the short term with the tools that we have or extensions of the tools. And that was one of the things from Mary Rinella's talk that I really took away, and that is that we're not great at that. We've got lots of traditional technologies, HVPG and elastographies coming, but really understanding how that predicts outcome in large populations is, I think, going to help us understand where we need to get to.
Yeah, for me, I'm going to do my best to really enroll into biomarker studies that help get us a little bit further towards transitioning away from biopsies. So, and I think there's some exciting new information being presented this afternoon about emerging therapeutic agents as well that are sometimes preclinical or early clinical. So those are my final thoughts, Roger. Thank you for having this wonderful podcast.
Thanks for joining. And so here's my final thought, which is that Donna and Louise and I have had these conversations, but we theme out this podcast in terms of what are the things we want to be looking at. And I feel a much greater need to focus attention on which patients need help most fastest, what are the things that will help them most fastest, and I take a holistic view on what that is.
You've been listening to the Surfing the NASH Tsunami coverage of NASH-TAG 2021. Have any questions for the surfers? You can post them on the Surfing the NASH Tsunami discussion group on LinkedIn or Facebook, or send them to questions@surfingnash.com, and we will address them as part of the NASH-TAG wrap-up episode. Thanks for listening. See you tomorrow for session number 4 on the Surfing the NASH Tsunami coverage of NASH-TAG 2021.